crossing over between genes C and D, but nothing again. And so there are regions of a chromosome where you get increased crossing over and regions where you get decreased crossing over. And in Drosophila, for example, along the X chromosome, if you take 4 genes, these 4 here, these 2 genes are 2 map units apart. These 2 genes are 2 map units apart. So when you sequence the Drosophila chromosome, you find these 2 genes are only 500,000 base pairs apart, and these 2 genes are 1,000,000 base pairs apart. So you can see there is a big difference between recombination frequency based on this distance, actual distance. So these 2 genes are undergoing a different rate of recombination compared to 2 genes. The same thing happens with humans. And this is an actual recombination rate along human chromosome 12. They measured the number of recombination events that took place, and they were able to map recombination frequency. And you can see there are peaks and valleys along this entire chromosome in human cells. And the reason you get variation in recombination along the chromosome is because the chromosome is not a single molecule of DNA that is stretched perfectly straight. It's actually folded. And because it's folded, you get a crossover. You're getting a crossover here to here, but you're not going to get a crossover between this sequence and this sequence, because we're far apart. Crossing over is actually a physical exchange. It only happens in the region where the 2 sequences are in close proximity to one another when the chromosomes pair. And there are numerous factors that affect this. One of the factors is proximity. The other is the chromatin structure is different in different parts of the chromosome. And that affects the formation of breaks, and therefore the rate of recombination. So we get the issues. So there was a study that was done in 2010, I believe. So in yeast, recombination usually occurs at promoters of genes because they are more open structures. And they reduce recombination of things like the centromeres and the telomeres. In humans, you get more recombination in regions that are genes, less recombination in regions between genes, but you get more often recombination in intergenic regions of those genes. So in the regions where there are lots of genes, recombination seems to occur more often in the regions between those genes rather than within the body of the gene. And the reason is that intergenic regions are where the promoters of the genes are. So in a sense, it's very similar to what we observe in yeast, that recombination tends to occur more often at promoters of genes. And it rarely occurs in regions that are very highly condensed. And there is, in humans, there's actually a sequence that accounts for almost 40% of the hotspots of recombination. It's the GC-rich sequence that takes place. So this was a study that was done in 2010 where they sequenced the genome of 4 individuals in a family, the father, the mother, and 2 children. And based on that, they were actually able to precisely map where the recombination events took place in the father and the recombination events took place in the mother. And what they found was, on average, there were 100 crossovers in the mother, but only 60 crossovers in the father. So that's very similar to what I told you about the sex-specific differences, that females have greater recombination than males. And then they also found that about 100 of these recombination events occur at what were originally thought of as hotspots of recombination. 60 of them occurred at random, but then 100 of those, 160 crossovers, occurred at those hotspots. All right, so I've just mentioned here that you get more often, you get recombination in regions between genes. But you do occasionally get recombination within a gene. So when we were mapping those genes, we were pretending that the genes were independent, and the recombination was only taking place in the regions between those 2 genes. But we do get recombination within a gene as well. And there was an experiment that was done which showed this. So until this point, we assumed genes were just sort of like dots. They were kind of unique and they were indivisible. Nothing happened to the gene, everything happened in the region between those two genes. And so they were indivisible functional units. And so genes we assumed were fundamental units of function. If you only had a part of a gene, it would not function. And they are fundamental units of mutation. When you change any of that, you basically destroy the gene. And that they were indivisible based on recombination. So I'm going to show you now is data that suggests that you get recombination even within a gene. So, how would you study recombination that occurs within a gene? How do you detect that recombination? So, when we are looking at two genes far apart on a chromosome, we are looking for the recombinant class, correct? You start with true breeding parents, and then you ask for the appearance of recombinant progeny that have shuffled their genes after they've gone through meiosis. So two genes in Drosophila, fork, which is a recessive mutation, gives rise to fork bristles, and carnation, which is a recessive mutation that changes the color of the eyes. So here is assumed fork bristles, just a cartoon diagram, and here is the carnation gene. And so what we're measuring, when we're measuring recombination, is we start with a parent with one combination of these two genes, and another parent with a different combination of these two genes, and we look for crossing over between those two genes. So, we start with a heterozygous individual, let's say a female heterozygous, crossed to a male. It's an X-linked gene. So here is the heterozygous individual, and it's this heterozygous individual that you're going to analyze crossing over. You really don't care about whether there is recombination in the male and Drosophila in this class. The reason you don't care is because it's an X-linked gene and the hemizygous chromosomes. So it doesn't matter if there is or isn't any recombination in the male. You're not going to do any multiplications by 2, etc., that some of you might have heard about from your TAs. I just wish you hadn't heard about it from your TAs, because it's such a Drosophila specific effect. I don't bring it up in this class. Your TAs confuse you, and then I have to clarify that whole issue for you. So it's unfortunate. But anyway, don't worry about it. In this, what you have to focus on when you're looking at recombination is a heterozygous individual. The only time you worry about Drosophila, males, and recombination, and then you multiply by 2, is if the male is heterozygous. There was a question in the class, in the midterm, where some of you multiplied by 2 because it was a fly question, not a Drosophila question, and you multiplied by 2 because you said, well, it's because there is no recombination in the males. I have to multiply by 2. If you read the question carefully, I purposely did that question the way I did. The female was heterozygous, the male was homozygous recessive. It's the last question, the long answer question. So you don't have to worry whether there is or isn't any recombination in the male. And that's something you have to remember. Recombination only occurs, and you will detect recombination only when they are heterozygous individuals. Recombination occurs in homozygous recessive parents, but you will not detect it. And so you just ignore that aspect. So when you do a test cross, you set up a test cross in a way such that you don't care about recombination in one parent, you only care about recombination in the other parent. So you don't have to worry about the multiplication by 2. So you guys lost some points because of that. In this example, again, notice it's the female that's heterozygous, the male is not. The male is hemizygous. Well, anyway, in this case, it's X-linked, so it doesn't matter. But if it was a classic test cross, you almost always take a heterozygous female, cross it to a homozygous recessive. So whether Drosophila has recombination or not is immaterial. So here we have recombinant heterozygous female. We're going to look for recombinant progeny. The male is going to produce two kinds of gametes, big F, big C, and Y, being hemizygous. The female is going to produce four kinds of gametes, big F, big C, little f, little c, big F, little c, which is a crossover event, and little f, big C, which is a crossover event. So these are your four gametes. What will this fly look like? It will be big F, big C, and so it will look like that. This fly again is big F, big C, little f, little c, so wild type. Big F, big C over big F, little c, wild type. Big F, big C over little f, big C, wild type. So all the females will look wild type. So you can't really say anything here. When you look at the males, you get big F, big C over Y, which is wild type. Little f, little c over y, which is forked incarnation. Big F, little c over y, which is only carnation. And little f, big C over y, which is only forked. So here are your two recombinant classes. Here is the wild type class. When you look at the numbers, you get numbers that are 400, 450. And so based on this, you'll say, okay, here are my recombinant classes, here are my parental classes. It's basically 100 out of 1,000, so it's 10 nanometers. Distance between genes. Approximately 10%. So you're looking at recombination frequency, you're looking at recombination between these two genes, and you're looking for this class of recombinant genes that are being generated. So what about recombination within a gene? So, recombination rates on average are a function of distance between two genes. So, greater the distance between two genes, greater the chance of getting a crossover. So, closer the distance between two genes, less chance of there being a crossover. So, for forked incarnation, the recombination frequency was 10% of the time. So 10% of the progeny underwent a recombination event because they are 240,000 base pairs apart, the two genes on the X chromosome. So because they're so far apart on the chromosome, there was a 10% probability of having a crossover between those two genes in a population of mice. So what happens as you bring the genes closer and closer and closer and closer and closer together? So, to find out a recombination that occurs within a gene, you have to have two different alleles of the gene, and you want to know if there is recombination between those two alleles of the gene. That's what you basically want. So here is what we are looking for. Here is fork, here is carnation, and we are asking for the change in the combination of these genes in the progeny. That's the classic method of recombination. Looking for a crossover so that little f goes with big C and little c goes with big F. As you zoom in and you bring markers closer and closer, if you were to look at recombination frequency between this gene and this gene, it would be very rare compared to here and there. And then if you look at markers that are very close within the gene, this frequency would be super rare. But it doesn't mean it's zero. And that's what you're going to see. How do you do this experiment? So, on the X chromosome, there's 10 megabase pairs, or 10 million base pairs. That's the size of the X chromosome in this. So the white gene, which we've been studying very often, is only 1,000 base pairs long. So it's a tiny fraction of the entire X chromosome. So to look at recombination within a gene, we perform an identical cross like we did before. We really don't change the cross. The only difference is we start with two different alleles of the same gene. So here is the white gene, W1, it's one allele of the gene. W2, it's a different allele of the gene. What are different alleles of a gene? They are sequences that are different in the same gene, correct? That's why you have two different alleles, two different forms of the same gene. So, imagine you have a mutation. Here is the white gene in this blue box. And here is a diploid fly. This is a diploid fly here. Here is a mutation in one copy, W1. And here is a mutation in the other copy on the other chromosome, homologous chromosome, W2. So this is a heterozygous fly with regards to W1 and W2. They're two different genes. It's a homozygous recessive for the white gene. It's W minus for both. But here is one allele and here is another allele. Okay, so we're going to look at recombination between these two alleles. That's what we're going to basically do, just like we were doing for the forked incarnation. So when you have a W1 mutation here, the sequence here is normal. When you have a W2 mutation here, the sequence here is normal. Right? So if we zoom in on the white gene in this heterozygous individual, W1, W2, there's a mutation W1 here, there's a mutation W2 here. We're going to look at the combination of crossing over that takes place over here. And to do this experiment, you start with a fly that is W1, W2. This fly is white, but it's a heterozygous white because it has one allele that's W1 and the other allele is W2. And you cross it to a big W1, just like we did with the forked and the carnation. Here is the heterozygous fly, and we're going to look at crossing over that takes place in this individual female fly. The male is going to look like this, big W and Y. The male is going to produce two kinds of gametes, the big W and the Y chromosome. The female will produce four kinds of gametes because of crossing over. One of them will be just W1. One of them will be just W2, and then two of them will be based on crossing over. So just W1, just W2, and then when you have a crossover right here, what will the gene look like? You will have one gamete that will be W1 and W2. What's the fourth gamete going to look like? It will look wild. It will look like big W. So, when we look at the progeny, in the female, it's not very useful. Everyone is red because the male is giving the big W chromosome, and so every fly is going to do it. So we ignore the females. Focus on the males. This one will be white, but it will be W1 white. This one will be white, it will be W2 white. The third class will be white. It's a combination of W1 and W2 white. And the fourth class will be red. So when you start to see red males appear in this cross, you basically know these red males are because of a crossover between these two alleles of the white gene. Now here, you do have to multiply the number you get for this class by 2. Why? Anybody want to hazard a guess why you have to multiply? Let's imagine I got 4 flies that were red eyes out of 100, or out of 1,000, or 10,000, whatever number. Let's imagine I got 4 flies that were red eyes. And I'm telling you, you now have to multiply that by 2. Why?
This is because the W1, W2 one is also a double.
This class is a recombinant class, correct? But it looks like the parent. You're not counting it, but you should be counting it to measure the distance between the two genes. This class is only half of the recombinant class, and that is why you're multiplying by 2. Nothing to do with males not having recombination and females having recombination. It has nothing to do with that. It is because this recombinant class is hidden. And so you just basically assume it will have gone through those recombinations, and so you add the numbers and multiply. So, when the experiment was done, they actually analyzed 10,000 males. So they analyzed about 20,000 flies. They sat there in the microscope and they actually measured 20,000 flies. Actually, they didn't do that. They did it differently. Quite fun. And they got 4 red-eyed males. And they multiplied that by 2 to get the distance. And it was 8 over 10,000 or 0.08 micrometers. So very, very tiny distance between those two segments. So here's the actual formal example. So you guys are now familiar with what a gene looks like. So imagine this is the normal white gene. This is a made-up sequence, but imagine this is the normal white gene. Okay? ATG, GGG, TTG, CCC, TTG, AAA. That's normal. So you'll get red-eyed flies if you have this gene in the fly. Imagine this is W1. W1 has a mutation, so it goes ATG, CGG, TTG, CCC, TTG, A. So that C is a mutation instead of a G. It's converted to a C. The W2 allele has a mutation. Instead of TTT, it goes ATT. Okay. So here are your two alleles, W1 and W2. So we made a fly that was heterozygous 1 and 2, correct? So we took a fly that was mutant 1, mutant 2, crossed to WY. The parental classes, the first parental class was this allele, ATG, CGG, TGG, etc. The second parental class was this sequence here, ATG, TGG, TGG, CCC, ATG, AAA. The third class was a crossover, and the crossover took place between this allele and this allele. Let's imagine the crossover takes place right here on the sequence. This is what the sequence would look like. You get ATG, CGG, crossover, TTT, CCC, ATT, AAA. That's the sequence. So we're crossing over between this G and this T. Coming down, we walking up. We exchange DNA. But it's reciprocal, so you have to go in the other direction as well. So when you go in the other direction, you go ATG, GGG, crossover, TTT, CCC, TTT, AAA. And this looks like the normal gene. So you've recreated the normal gene. Right? And that's why you get the red-eyed male flies, the 4 red-eyed male flies that you found. Okay, so this is known as intragenic recombination. It is the recombination that occurs within a gene, while intergenic recombination is recombination that occurs between genes. It's very rare, but it does occur. And because recombination is so accurate, you don't lose a base pair, you don't gain a base pair, you just basically replace one strand of DNA for homologous chromosomes. All right, so that's the entire lecture 8. We'll take a break, and then we'll come back and continue.